I know is that is after my child's bedtime, which is the only kid-free time that is guaranteed for me. So if I'm going to, at the moment, that's right. That's my prep time for this class. Like last night when I needed to prep for this, I was reading it up. I'm really happy that tomorrow's journal day for you guys. First off, any questions on journals for tomorrow?
Yeah. So, like, what range of topics do you have to choose from?
Okay, so your topics are any of the chapter titles. So look in our book, see what chapters there are, find one that's interesting, and look up an academic journal on our school's website, like the library. There's a journal article, and make sure you put like, somewhere you have to put advanced search and then like peer-reviewed journals only and full text. That way you don't have to like click on it and try to figure out how to get it. Yeah, the book is online somewhere. Say, or find a friend.
On files for the presentations, the PowerPoints, it goes to chapter 2 and chapter 5. So chapter 3 and 4.
We are not covering those in this class. There's too many chapters for how many class periods that I want to lecture in, which is bare minimum. I much prefer doing things outside, and this is probably the most lecture-heavy class that we're going to have in this entire major. I would prefer it to be that way. I don't know if I set it up that way because this was the first class that I did this with last year, and I didn't get as good at it as when you took like motor learning and biomechanics. But it is the same concept that I would prefer, but I'm trying to decide how we want to do that. Here's what I think I'm going to do tomorrow. We are going to do your presentations alphabetically. So if you're early, you have to prep the same way. So we will do as many as we can. So you need to be able to present in 5 minutes. The three big questions, what happened in it, hypothesis, conclusion, how did they test it, what were the test subjects, did they prove it or not, then how does it apply to this class, how does it apply to you. So you need to be able to talk about that for 5 minutes at least.
But we don't have to have anything up there like that.
I'm not going to require it this time because we have so many, because then we're going to get bogged down by, unless you've got stuff that makes it more interesting, that's what I'm going to say. If you've got it, put it, when you turn in a PowerPoint that has, or even if somebody used to do PDFs, they would make a PowerPoint and then turn in the PDF of the PowerPoint. I'm not sure how he did that. But that's something that if you just have like a graph or a chart that would make it more interesting with what you're saying, that's going to be make it more interesting for us as well. So you just sitting there talking is not necessarily the most interesting thing. So that's why eventually we're probably going to make the requirement to make a PowerPoint, but I want to see how many presentations we can get through in a 50-minute class period. So it's going to be alphabetically. So if your last name is Early, you're probably going to be talking. I can see it's like, oh.
So we'll go over to Friday.
We don't have class on Friday.
Oh, I mean,
it won't. No, we'll just however far we get alphabetically. Next Thursday will be another journal, and we'll start at wherever we are in the alphabet is the first person to present next week. And we'll just keep working our way through and looping it back. And for the 4 journal presentations, which I think is the first 4 Thursdays.
So every week, everyone still has to do a journal?
Yes. You may or may not present. But you still need to be prepared. That's why I don't want to say, these are the only people that are going to present, because then half the people aren't going to make it. But, I mean, if we can do it in 5 minutes each, in 50 minutes, that's 10 people should be able to present, which would be most of you. If we can actually keep it to 5 minutes. And some people might go long, some people might go short. So that's why I'm not going to make some go crazy on this one, on making sure you have a PowerPoint or not. I prefer it with a PowerPoint because it's more interesting to see it rather than to just hear you talk about it.
So as long as we hit all the points that you told us to, like the length of our pH?
No, no. As long as you cover it all, just make sure it's APA format. So that means like double space Times New Roman, has your name, date, class number, professor, title. There's a rubric to give you all those details. Any other questions on that? Because I'm probably going to be yakking the rest of the time because there's another lovely 71 slides and 2 videos. Ooh, I did not put the right amount of time on there. I added questions to it and didn't change the amount of time. So I have to grade those tonight or sometime. Usually Monday morning is when during my weight training class is when I end up grading because it's the only kid time that I don't have to deal with things. So most of the time I say Monday morning is my getting grading done day. But yeah, I will grade those at some point. I will try to do it sooner because you're going to take another one tonight. So I will. Yes, I will make sure I have the same amount of minutes as questions.
Yeah, that was like 35 seconds per question.
And you have to type in one of them too.
Yeah, you have to type 2 of them. Yeah.
Some people, I think there's 13 of you. I had 2 of them that were said graded and 11 that said needs grading. So it means 2 people got lucky and didn't have to type anything. So some of it is luck of the draw, some of it's not. All right. So this chapter I didn't do last year, but I realized we talk about this so much. And then we really don't talk about it until your senior T&T of weight training and development. We'll talk about this again. And in even more detail, like it splits into 3 different days. So that's why I made sure we went over this because we refer to this a lot. So I'm probably going to go fairly quick through this to go through that. So we're going to talk about how the body gets energy and all the different systems that you go through as you work out or as you exercise. So some of the basic terms here, there's only 3 today, lucky us. So you have your substrates. So those are the fuel sources, which the main one you're going to have is ATP. And we're going to talk about how carbohydrates, fat, and protein are broken down into ATP for you to use for your body to make those actin and myosin things get together and make your muscles move. So the bioenergetics, so this is all things done at the cellular level. And your metabolism is all the reactions in your body. You hear people say they have a high metabolism, a low metabolism. The younger you are, the higher your metabolism. The older you get, the lower your metabolism gets. Tragically, it's true. All right, so measuring the release, everything is calculated from heat. So a calorie is actually a heat unit. It is not a true, maybe you've taken nutrition or not, but it is actually a heat unit that is required to take 1 gram of water from 14.5 Celsius to 15.5 Celsius. So that's how they calculate it. So 1,000 calories is a kcal, and that would make 1 calorie that we do in a diet. So the main things that you get this from, and this is good old health class right here, we'll go quick through it, is carbohydrates, fat, and proteins. Those are broken down into carbon, hydrogen, oxygen, and nitrogen. And the energy from most chemical bonds is stored in the compound called ATP. And let's see, when you're resting, 50% is carbs, 50% fat, but you're not using very much there. Then your quick exercise, you're going to be using more carbs. And when you exercise for long periods of time, that's when you burn through your carbohydrate reserves and start actually burning fat. So that's why they say if you want to lose fat from your body, you need to run or get your heart rate up for a long period of time, more endurance things. When you're doing short things like lifting weights, it's going to be all carbs. So why do we want to cut carbs? We'll hit that one another day. Oh, here's the ones that are from our old. So carbohydrates have 4.1 kcals per gram. and your body can store 2,500 of them. Your glycogen is converted to glucose, which is what is needed to make more ATP. Your glycogen stores, it's about the same here, is your dietary carbohydrates need to be replenished each time. And you'd have to burn through those to get through to your fat reserves so that you can burn fat. Fat has 9.4 kcals per gram, and you can store 70,000 plus, actually it's unlimited really, how much your body can store. Energy substrates prolongs and with less intense exercises. So the fat-free fatty acids are used to also make up ATP, and we'll go through that in the video here in a little while. So carbohydrates are stored in the liver, in the muscles, and in glucose. And you can see how much are stored in which area. Your fat locations are subcutaneous, under the skin, and visceral, around the organs, in between the muscles, intramuscular, and in the glucose of the body fluids. And that's actually where a lot of transport is done in the body, is with those fat in body fluids, so in your blood and those kind of things. Protein also is 4.1, which is the same as carbohydrates, calories per gram. And also it goes through a different thing called gluconeogenesis. This is one that's going to use a lot more of these free fatty acids, also to make your energies get stored. So if you were, I had this one in college in my health class, you're stranded in a car in the dead of winter. What do you want to eat to last the longest? Fats, because it has the most calories per gram. Good. All right, so this is a very quick way. So you eat, you have your fats, carbohydrates, and proteins. Your fats are stored as triglycerides, and they take free fatty acids and glycerol, and then those go through lipogenesis, and they eventually turn into the metabolism, which is your energy. Carbohydrates get broken down into glucose. Any excess, so if you eat too many carbs, it gets turned into fat. So any excess from that glucose process goes into fats, which turns into there. And that goes through your glucose and glycogen, so that energy process there, which there's a little bit that goes that direction, not much. A little bit of amino acids there coming there. So proteins, it has a totally different breakdown. Your body's proteins and all those amino acids that you have to eat. That's why you want to eat stuff that has beans and rice, has a complete protein because it has all the amino acids. Also, meat has all the amino acids too. But we're at an Adventist school where we talk about a lot of vegetarian diets. All right, so controlling the rate of energy production by the energy that is the substrate that is available. So the energy released at control-based rates are availability of the primary substrates. Substrates is going to affect the metabolic rate. More available, the higher pathway of activity, the less excess of given stuff, cells start relying on the substrates more than others. So energy is released at a controlled rate. Did it literally say that on the same thing? Nope, slightly different. Based on the enzyme activities. So the enzymes do not necessarily start the ATP yield, but they do facilitate the breakdown of all these substrates. So the carbs, the fats, the proteins, enzymes facilitate it, they don't actually start it. The lower the activation of the energy, and that's where you end up getting that suffix ase. So enzymes are added to ATP, and that's what helps break down. ATP is broken down by ATPase. So anytime you see that term there, that's what they're talking about. Specific enzymes are required for each step. So the more enzyme activity, the more product is going to happen. So there is some limiting to that. There can be a bottleneck at the beginning, which you're going to see in a chart in a second. Activity is influenced by the negative feedback, and this slows overall production. So you have your useful energy, phosphocreatine. Anybody heard of creatine before? This is why people eat creatine when they work out to try to get bigger muscles. And it is one of the actual few things that is okay to actually eat, but we'll talk about that another day. Phosphocreatine, so you're going to have your no enzymes is when it starts going, and then the creatine ends up being down here. So phosphocreatine, chemistry happens, you'll get some activation energy, and then it turns into creatine. And some enzyme ones, it's not going to go quite as high, you're not going to get as much energy, as it turns into creatine for your muscles to use. So yeah, we're going to watch a video about this. Let's just go straight to it. Oh boy, is this going to work?
Chemical reactions occur only when the reacting molecules achieve a sufficient initial energy state to start the reaction. In metabolic pathways that form adenosine triphosphate, or ATP, an input of energy in the form of stored ATP is typically needed to begin the series of reactions that ultimately release energy. One can think of this as priming the pump. Enzymes present in the pathways control the rate of energy production, often speeding up the reaction by lowering the activation energy needed to begin the reaction. Biochemical pathways that result in the production of a product from a substrate typically involve multiple steps. Each individual step is typically catalyzed by a specific enzyme. As fuels are subsequently degraded into byproducts along the metabolic pathway, ATP is formed. Utilization of the stored ATP results in the release of usable energy, heat, and the release of adenosine diphosphate, or ADP, and inorganic phosphate. Enzymes can be inhibited through negative feedback of subsequent pathway byproducts, slowing the overall rate of the reaction. This usually involves a particular enzyme located early in the pathway called a rate-limiting enzyme.
And that's another one. We'll talk about that rate limiting stuff later. Yeah, moving along. And that guy talks. Standing break time. We've been sitting for about 15 minutes. Woohoo!
Every time you hit legs now, force myself to go really heavy squats.
You want?
It says, it says like the highest jumpers, they're like squatting over double the body weight.
Probably.
Yeah, so I'm trying to squat a lot more than double the body weight.
But that's going to be like their one rep max is double their body weight.
No, I'll do like 2 or 3 reps.
Okay, so that's not that much more.
Yeah.
I mean, that's still like
it's double. It's like double your. All the ATP stored.
it's the one that you're, all those carbs you're burning up immediately. So that's what we're going to get to.
ATP. We love it. You'll find ATP in so much of our science art.
All right, you got.
Whether it's
This is as loud as I can get it today,
mitochondria or
guys.
cell transport, or a comic about cellular respiration or fermentation, you'll see ATP mentioned. So why the big deal? Why is it all over the place? Many times students will get it in their mind that it is an energy currency of some kind. When I first started studying biology, I noticed that in textbooks, it's often represented like the starburst thing or thunderbolt. And, I guess in my mind, I imagined it was like some big blast of energy that helped the cell do things. And by do things, I mean that we need ATP to do many cellular processes. Examples include active transport, such as when a cell is trying to move something against its concentration gradient, or its role in muscle contraction with the actin and myosin cross bridge. We need another video for that. ATP is critical for many types of cell signaling. You need your cells to be able to communicate. Those are all just some examples. But what is ATP? How do we get it? And how does it work? Those are the basics of what we're going to focus on in this short video. So what is ATP? If you remember the 4 major biomolecules, ATP would fit in with nucleic acids. Yes, like DNA and RNA. ATP is a nucleotide derivative, so it has those 3 parts you'd see in DNA or RNA nucleotides. Phosphate, sugar, base, but it actually has 3 phosphates. ATP is short for its full name, adenosine triphosphate. This fancy name is helpful as it tells you that it contains the nitrogenous base known as adenine and 3 phosphates, hence the tri in adenosine triphosphate. Its sugar is ribose. How do you get ATP? All cells need ATP, and so they need processes that can be used to generate it. But the process can differ. It might involve oxygen, such as aerobic cellular respiration. It might not involve oxygen, such as anaerobic respiration or fermentation. During cellular respiration, plants break down the glucose they made from photosynthesis to make ATP. During cellular respiration, animals break down the glucose they consumed to make ATP. And it's not just plants and animals. Bacteria, fungi, protists, and archaea, they all need to make ATP. We have a video on cellular respiration and another one on fermentation that can be helpful to understand the process. But one thing that we do want to mention about making ATP is it's important to understand it is part of a cycle. With the ATP cycle, you have ATP, which can be hydrolyzed, releasing energy and losing one of its phosphates in this process. A process like cellular respiration can provide the energy needed to add a phosphate to ADP in order to regenerate ATP again, which is important as ATP can be used quickly. This brings us to how ATP is able to work. So how does ATP work? It's not just about ATP being hydrolyzed and releasing energy, it's more than that. Okay, honestly, it's more than our short video can go into, which is why we provide some further reading links, but let's look at some basics. So when ATP is hydrolyzed, meaning here it involves the addition of water, it's not really that the bond between the second and third phosphate itself is a super strong bond. It's actually more that the bond between the second and third phosphate contributes to this ATP being unstable. These phosphates with their negative charges don't like being arranged like this. The change from ATP, losing its third phosphate, to become the more stable ADP is an exergonic reaction and releases free energy. A popular example for understanding ATP is to use the spring illustration, like a wire spring. Consider how you might compress the spring. ATP would be modeled by that compressed spring, and then you would let it go until it just goes into this relaxed state, which would be represented by ADP. When ATP is hydrolyzed, if the energy was just released, it will likely not be useful for a cell if it's not actually coupled to something that needs it. Thankfully, the energy release can be coupled to endergonic processes that the cell needs to do. This can occur when the phosphate from the ATP is transferred to a molecule that is going to be acted upon. For example, this cell transport protein here is supposed to move some kind of molecule against its concentration gradient. Recall that if it was passive transport, these molecules would be moving from high to low concentration. But in active transport, thanks to ATP, this protein can move them against the gradient. When the phosphate is transferred to this protein, we say the protein has been phosphorylated. Sounds powerful. We can say in our example that this protein is more reactive and less stable in this form, this phosphorylated intermediate state. When it reverts into its original, more stable shape, it can assist in moving them the other direction. So, from marveling at the beating of a single cilia hair, or chromosomes being separated in cell division, or binding the correct amino acid on the tRNA, I could go on. We hope that little ATP symbol will mean something every time you see it. Well, that's it for the Amoeba Sisters, and we remind.
All right, so that's some actual graphics of what we're doing here, which I think is easier to understand than looking at like chemistry symbols, even though most of you are either in or have been through chemistry at some point. So ATP stored in small amounts until it is needed. It breaks it down with ATP, water, and that enzyme ATPase into where they drop one. So basically what they were just talking about there. Then the energy goes backwards and it goes back into, uses energy and makes it into triphosphate again. This can occur either with the absence or presence of oxygen. So you do still do these reactions whether you're doing aerobic or anaerobic exercise. And there's another wonderful chart of the exact same thing that we kind of talked about there, which I think their example is a little easier to understand. So biogenics, you have limited ATP storage. It constantly is synthesizing new ATP, and there are 3 pathways that you're going to use, and you use these kind of in order to a degree. You always start with your ATP to PCR system, and I have a video about these in a second. So that would be your anaerobics. So when you first start running, you are using that system first. That's the fastest one that you're going to use up that ATP. And then they're going to be trying to react and make more of it. Then you start the glycolic system, which will be from like your 15 seconds to a minute is when you're using that one. So if you're in one volleyball match there, you're probably using these anaerobic systems for the majority of each play. Or if you're playing football, you're probably only using this system. And the more tired you get, the faster you go through the first system and start working on the second one. So like when the defense has to stay on the field in football for too long, all of a sudden, now they're only using this for 1 second of the play instead of the entire play, and that's how they're getting more and more tired by the fourth quarter, and why the offense can finally get a running play to work in the fourth quarter. It's because they're tired. And eventually you get to the oxidative system, which is the stuff that you use when you're doing distance running. So anywhere between a minute to an hour plus, however long it takes to run like a marathon, is when you start using mostly that one. And the percentage changes as that first minute to 3 minutes goes along. And this one, they will explain it a little bit better.
Energy is needed to produce physical movement, maintain body temperature, and keep up with all the metabolic activities in the body. In our bodies, all physical movement relies on the compound adenosine triphosphate, or ATP. ATP is stored in the muscles, so it is immediately available to produce movement. However, storage of ATP in the muscles is limited, so any movement that lasts longer than a few seconds requires more ATP to be produced. Our bodies have three systems that can produce ATP: the ATP-PCR system, the glycolytic system, or lactic acid system, and the oxidative system. The first two systems are anaerobic systems, meaning oxygen is not required to produce ATP. And the oxidative system is aerobic, because oxygen is needed for ATP production. The ATP-PCR system is based on movements lasting about 5 to 15 seconds, such as strength power movements like a golf swing, jumps, throwing, or racket serves. These type of quick burst movements can be done with ATP that is stored in the muscles. During short sustained power movements, or any quick bursts such as a short sprint, ATP will provide the energy alone for the first few seconds, with a compound called creatine phosphate, or PCR, offering the ATP for another few seconds. Like ATP, the amount of PCR is limited, so this system can provide energy for movements up to about 15 seconds in total. This energy system produces ATP very quickly, but not over a long duration. Next, energy demand shifts to the glycolytic system. This system relies on the rapid breakdown of carbohydrates. Glucose, which is one of the most basic forms of carbohydrates, is constantly circulating in the bloodstream, and glycogen is a stored form of glucose in the muscles and liver. This blood glucose, or glycogen, is broken down to create ATP through a process called glycolysis. During this energy supply process, a substance called lactate is formed and hydrogen ions are released. It's believed that the accumulation of these hydrogen ions in the muscle causes the muscle to become more acidic, contributing to fatigue and a burning sensation. Exercises that are performed at maximum rates for between 1 to 2 minutes, such as a 200-yard dash, depend heavily on the lactic acid system for ATP energy. This energy system produces ATP very quickly, but again, not over a long duration. The oxidative system involves the use of oxygen. Through two complex metabolic processes, the Krebs cycle, or citric acid cycle, and the electron transport chain, ATP is produced. This energy system, with all of its processes, can't produce ATP nearly as quickly as the first two systems. However, this system can produce ATP for a much longer duration. This system can use carbs, fats, or if necessary, even protein. However, fats and proteins have to be broken down, so when the system uses them, the process becomes longer. This system is used for longer duration activities, such as a bike ride or long run. The chart on the screen shows an estimate of the percentage of energy used from each system, along with...
So that's a really good chart there of which system and how long. So that you start out really using that PCR system first, and the glycolysis system just kind of goes up, and eventually they swap. Your glycolic system eventually does taper off, and then the oxidative system pretty much takes over. But the glycolic system doesn't shut down like the PCR system. Eventually, what's already in there is gone, and the rest of the time, these other two are basically trying to reset how much ATP you have in the muscles. I just really like that chart, and we'll have something similar down the
with
road.
time plotted at the top.
Probably it's going to be just the glycolic system. That's why it's called anaerobic. So when you fail, I mean, unless you're doing, I'd say my class here was doing 25 reps for the first couple of weeks here. 25 reps, you might be going for 2 minutes, depending on what, if you're doing 25 squats with fairly light weight and you're just trying to get the control of it and learn the motion, they might be getting into the oxidative system there, but probably not. So I'd say you're probably in that, if you're going to failure, your first way you're going to fail is the PCR system. Then you're going to fail when the glycolic system can't keep up with it, and that's when it fails. So it would probably be closer to like this point is that failure moment, is when this one can't keep up and this one's done, that's when your muscle fails. Make sense? Good question.
As you can see, the ATP PCR system dominates at the beginning. The glycolysis becomes the dominant energy provider, and then energy really relies on the oxidative system. Here are 2 other charts. The one on the left shows an estimated rate of ATP production per second for each system. It also shows the rate for carbs and fats for the oxidative system. You can see how much more rapidly the ATP, PCR, and glycolysis systems are at ATP production. On the left is a chart showing the aerobic ATP production for trained and untrained people, showing the benefits of well-trained persons as they don't run out of energy easily. Each of these systems can be trained to be more productive. By changing your workout routines, doing explosive moves like box jumps, you can improve System 1. By doing circuit routines, going from one exercise to the next, with little to no rest in between and using lighter weights, you can train System 2. And by doing a 20 to 30 minute cardio session of low to moderate intensity, like walking, jogging, or biking, you can improve System 3. It's a good practice to train all 3 systems by altering your workout routines. The body is amazing, and it adapts very quickly. Changing up routines also provides mental stimulation, which helps overall health. It can help people stay energy.
We're going to keep going on there. That is one of the most interesting things, is you can train which system you want to work. And that's why coaches in like football and volleyball are going to do a whole lot more sprinting than they're going to do, because their plays last a certain amount of time, unless you have like a crazy one like the Nebraska game the other day where they went over a minute. Oh, that was nuts. Side note. But yeah, you can train all those things, and this is I'm gonna really skip over these because we pretty much just talked about these and how long they do, and see if there's anything other. There's all the chemistry that goes with there for those, how much time. There's that phosphocreatine to creatine, and that creatine is the same thing that you would actually, I think most of your creatine powders are actually closer to the phosphocreatine than the actual creatine, but it's really hard to say that, so that's what they sell. So these would be those systems again, and there's your exhaustion point after 15 seconds. Your ATP and the phosphocreatine reaction going as those fail, and that's where your muscles are just done at the end of the lift. These would be your fast twitch skeletal things, as I have a note here for that. So your type 2 fast twitch muscles here is where this is all happening. All right, PCR breakdown by the creatine kinase. I'm not good at pronouncing things, in case you haven't noticed. So the creatine kinase controls the ATP production in a negative feedback loop. As the ATP drops, ATDP goes up, and so does creatine activity. When the ATP levels go up, the creatine kinase goes down, and these are all inside there. All right, standing break, and turn on the lights.
That's all I have for videos.
We should be like, run the stairs, up and down, and come back. Dude, get a job at a golf course, then you get free golf.
That's what he did. Kind of be a groundskeeper, just so you know.
Oh no, I wouldn't want that. You actually have to have a passion for.
Oh my gosh.
There is a full associate's degree on groundskeeping. for golf courses.
Oh,
There's
okay.
2 years of education to do that.
That's my top thing, just to see how it goes.
Now that I will agree that does sound fun. All right, keeping going. So some more details on here, and I'm going to go pretty fast since we've had 2 videos on it. So your anaerobic has a yield of the ATP yield of 2 to 3 per substrate there. It lasts at 15 seconds to 2 minutes. So that breaks down through glycolysis. So your glucose or glycogen substrate, depending on where it comes from, must involve the glucose 6. So this costs 1 ATP and 0 for the glycogen. And these are just a bunch of numbers here. I don't know if you really need to know all of them, but just knowing which one's more than the other is what you're really focused on in these slides here. So the yield for a glycolic system through using this glucose is going to be 2 ATPs and 3 ATPs for glycogen. Notice up here, it costs 1 to yield 2. So that's where you're kind of got to go. So some of the pros of this, it allows muscles to contract when oxygen is limited. So that's why people hold their breath sometimes when they are lifting. Don't do that, by the way. But some people do. Well, that's another one we'll talk about in TNT of weight training. Low ATP yield, lack of oxygen brings lactic acid, which is that burning sensation in your legs. Oh man, there's a fun word there that I'm not even going to try to say. So ATP goes down, ATP goes up. This activity goes up. So same with like that CK thing from a little bit ago. Usually less than 2 minutes. Then you have your oxidative system. So there's a little more chemistry here. 33 to 32 with 1 glucose adds 100 plus per 1 free fatty acid. This is usually things that are using the mitochondria, not the cytoplasm. So those mitochondria that are just there to make energy. Speaking of mitochondria, so the density within the mitochondria is determined by demand. So the location also determined by where the oxygen's coming from. And there can be multiple mitochondria in each muscle. So each muscle fiber. So oxygenated carbohydrates start first with the Krebs or the glycolysis, then they'll use the Krebs cycle, and then the electron transport as you continue to run farther. So Pablo ends up getting all the way to his electron transport chain as he's doing his super long runs or bikes or trying to climb his bike up that crazy hill by my house there, which I still don't know how you do without an extra energy thing behind you. And so here's kind of where those go through. You have cycle 1. So we had glycolysis, Krebs, and electronic transport. So your first cycle is here. The second one's here. And we could go through an entire class period on the Krebs cycle. Like, man, there's 20-minute videos on that. So just know that it's going to use carbon dioxide and oxygen. Are we going to go into all that detail? Oh, we might. So oxygenation, glycolysis occurs with or without. The ATP yield is the same, but in the presence of oxygen, acetylcholine, and Krebs, it enters the Krebs cycle. Do we have a video on the Krebs cycle? We might. Oh man.
In the Krebs cycle, the acetyl-CoAs that we made during the link reaction will get further broken down. Remember, these acetyl-CoA molecules are the remains of our original glucose molecule that we started with at the beginning of respiration. By the time we're done with the Krebs cycle, our glucose will be completely oxidized and broken down to carbon dioxide. Let's check out how the Krebs cycle completes this breakdown. First, acetyl-CoA reacts with a 4-carbon molecule called oxaloacetate. This makes a 6-carbon molecule called citric acid. For this reason, the Krebs cycle is sometimes called the citric acid cycle. Citric acid gets oxidized, transferring electrons and hydrogens to NAD+, forming NADH. This oxidation reaction causes the release of CO2 as well. Now we have a 5-carbon molecule. This molecule undergoes a similar process of oxidation, producing another NADH and another CO2. Now we have a 4-carbon molecule. This 4-carbon molecule undergoes another chemical reaction that transforms it into a different 4-carbon molecule. The energy released by this reaction is used to join ADP and phosphate to form ATP. Our new 4-carbon molecule gets oxidized even further, transferring electrons and hydrogens to a different electron carrier called FAD, forming FADH2. FADH2 will serve the same purpose as NADH, carrying electrons to the electron transport chain. The new 4-carbon molecule that results gets oxidized one more time, forming even more NADH. This final reaction also results in making our starting molecule, oxaloacetate, once again. This is why the Krebs cycle is a cycle. We end up right where we started. But notice that we made a bunch of useful molecules along the way. Perhaps most importantly, we have produced many electron carriers, NADH and FADH2, that will carry electrons to the electron transport chain. We also made ATP that can be used as energy, as well as carbon dioxide that will be breathed out when you exhale. Since we have one more acetyl-CoA that needs to go through the Krebs cycle, let's watch the process one more time with a slightly different perspective that shows its cyclical nature.
Alright, so that's all that chemistry that you see right there. All was in that video. I think the video is way more interesting, so we're just going to fly over those and make sure there's nothing else. And there's that whole chain and a big picture there. Alright, so after it goes through that process, then the electron transport chain happens here. So these hydrogen and electrons carry all of those NDHs and FDHs, which are the results there. Those travel down the chain, combine with oxygen, and those electrons and oxygen help form ATP, and then you get 2.5 ATPs per NDH and 1.5 ATP per FD. So yeah, all these things seem to help add more and more ATP that's going back into your muscles to help them contract, using the actin and myosin from yesterday. So the yield of one glucose with all that chemistry is 32. One glycogen is 33. I think if I remember right, that's a quiz question that's in there somewhere. Just a heads up. So the breakdown net totals, these you don't really need to know. Just know that there's a lot. That's where they all come from. You add all those together and you get one of those two numbers. All right, so this would all be done in that mitochondria in your cells. Say all this, you go down to like cell size, then you have like the mitochondria. Inside that little mitochondria, a little part of your cell is this going on. And they say there's multiple ones of these in each cell of your muscles, or each muscle strand. There's more than one. We could watch that whole thing, but I only got 5 minutes and I need to make sure there's nothing else. We already kind of saw that. Going on there. Krebs cycle, all these things, all the electron trains end up having, and here's that whole yield of 32. So just another way of showing that. And we could have another one there. Don't have time for this standing break. Oh, there we go. I like it. So it says the guy with way too much energy over there. Triglycerides. So when you start oxidating fat, triglycerides are our major source. Using free fatty acids, 3 to 4 times more ATP than glucose. So we were all the things we had talked about with that Krebs cycle. That was just the carbs. Now we're talking about oxidation of fat, and I'm not going to have a lot of time to do this one here. This is a slower process than glucose, which we already kind of talked about. So beta oxidation of fat, so using those free fatty acids, acetylcholine before entering the Krebs cycle, which was earlier in that first slide where we said some things end up being shot over from fats into that direction. Yep, so moving along there, so 16, a whole bunch of chemistry going on down here. Fat oxidation requires more oxygen and yields more ATP later. Part of the reason you get tired when you keep running or keep doing whatever exercises you're doing. And that Krebs cycle helps with that electron chain and sends it back to the Krebs cycle, and then it keeps circulating around there. So here's kind of those numbers there. So fat oxidation has a -2 of the oxidation. Then you quadruple it, and then you can go 8 times of it to end up with 106 more things there for your ATP production. There are different types of fatty tissues in your body. There is the white adipose tissue. These ones will have the lipid storage and breakdown. The brown tissues transfer energy from food directly into heat, and they may contain little droplets of these things into your blood vessels. Protein would be the other way that we can get it, or we can get energy. So it's a rarely used substrate, pretty much only when you're starving. If you have no fat left in your body, then you start going after proteins, which actually will start breaking down your muscles to give your body energy. And that's why when you see those starving children things, they don't do those on TV nearly as much as they used to, because it was pretty disturbing to watch kids with a giant belly and arms that looked like they were just bones. And that's because their body was using proteins to feed them because these kids were starving. So energy yield is not easy to determine, and it's just because it can't be studied, because to study it would be very unethical to purposely starve people. Lactate is also pretty important there, and they kind of already talked about that in that video, so we're going to keep going. Say lactic acid kind of goes in and out of the bloodstream there to where get that all, and there's some more chemistry going on. All three things. Oh, there we go. How much ATP is happening in each one as it works its way down. So that's what I say. But this one's sustainable. These are not. And that's how much energy. Say there's no limit at the end of this one per thing. And then here's all the chemistry numbers that happen in there and how long it takes you to get to that point. Last big one here is there is a part, so when you're exercising at 60% of your VO2 max, pretty much fats are going to do most of the excess. So there is a point, and I actually talked to a guy from Southern that his whole life's work in a lab similar to ours up there, he had a lot more computers in there and a couple more bikes and things. But he actually could take your VO2 max and that entire test, and he could say, you need to exercise at 61% of your VO2 max, and that is the point where you are going to be burning the most fat in your exercise. So he could tell you, you need to exercise for 20 minutes at exactly this, and you're going to have the best results. Like he wrote several papers. They were like on the walls of the department there at Southern for this guy. I'm like, wow, and he was retired. I'm like, okay, I'm glad I had this conversation now. But that's what this whole thing is talking about here, is you can find that spot, that crossover, where you're going to be using the most energy from fats and carbs. So carbohydrates goes up, fats going down, and you want to hit that point. And when we do our VO2 maxes, there is an app that goes with the thing that's there. You have to pay for it, though. I'm going to do it one of these days and then actually try those workouts, because it will actually give you kind of that same formula of where you can exercise for the amount of time and lose the most weight to burn the most fat. I want to try it because I'd love to get rid of that a little more. Wow, only got 4. Let's make sure there's nothing else here we need to know. Not all muscles are going to have the same oxidative capacities, depending on how big they are and things like that. Enzymes, there's all those restricting things there. Type 1 fibers have more oxidative capacity. That's why you can do it for endurance training longer. Endurance training is going to have, say, more type 2 fibers, has more mitochondria, more enzymes, and more mitochondria. Last slide. ATP demands increase with intensity. In response, the increases occur. You're going to need more ATP, more oxygen delivered by the heart and blood cell. Those O2 storage is limited. You either use it or breathe it out and you lose it. O2 levels entering and leaving the lungs enable accurate estimate of O2 use in muscles, and that's why we do the VO2 max test that you're all going to eventually do. I'm done. Made it through 71 slides and 3 videos. I had to skip a little bit there.